In this video, we will be diving into a fascinating topic and one that is crucial to understand in the field of medicine, the tetanus toxin. This toxin is produced by the bacterium Clostridium tetani. This powerful neurotoxin can lead to severe muscle spasms and can be even life-threatening. But how does it work and what can we do to protect ourselves? Well, let's get right into it. Let's first explore ways and how this toxin can enter the body. One common route this toxin can enter the body is through deep puncture wounds, such as when a person steps on a rusty nail. This can introduce C. tetani spores. These spores are highly resilient and can survive in harsh conditions, including extreme temperatures and lack of oxygen. Nextly, animal bites from dogs or cats can also lead to contaminated wounds. Traumatic injuries like fractures or deep cuts can also pave the way for Clostridium tetani spores to enter the body. Post surgical infections can occur after procedures like orthopedic or abdominal surgeries if the surgical site is not adequately cared for. Another way of entry is grass on migration. Humans can get grass awns lodged in their skin while hiking or gardening. And lastly, some individuals could experience cryptogenic tetanus, which means they could have developed the disease without a clear history of the wound, possibly due to environmental exposure to the spores. Now you've seen the possible ways this toxin can enter the body. What happens next? Well, let's take a look at where this toxin goes once the body has had exposure to it. Picture someone stepping on a rusty nail containing C. tetani. After germination of the wound site, the bacteria Clostridium tetani multiplies and produces a tetanospasmin toxin in an anaerobic environment, which begins to disrupt normal nerve function. This multiplied growth is often associated with deep puncture wounds where oxygen levels are low, creating a conducive environment for the bacteria. As the bacteria proliferate, they can cause local tissue damage as C. tetani secretes various enzymes such as proteases and phospholipases that can break down cellular components. This damage can lead to increased permeability of the cell membranes in the surrounding area and produce an inflammatory response. Inflammation causes the release of mediators that can alter the permeability of endothelial cells and surrounding tissues. Also, during this time, the bacteria are germinating in the wound, and this is when they release the tetanospasmin toxin. Due to the increased damage and increased permeability allows molecules, including the toxin, to leak out of damaged cells into the extracellular space. This gradient can drive the diffusion of the toxin into the surrounding tissue and eventually into the bloodstream. Once in the extracellular fluid, the toxin can be absorbed by nearby capillaries or larger blood vessels. The thin walls of these vessels allow small molecules, including proteins like the tetanospasmin, to pass through, and this ultimately facilitates entry into the bloodstream.
After the tetanus toxin enters the bloodstream, it binds to receptors present on sensory nerve terminals and enters the peripheral nervous system. The specificity to enter PNS neurons over others is due to their localization near the site of infection and the nitrogen coating around nerve cells, which attracts the tetanus toxin to them. To enter the neurons, the tetanus toxin binds with gangliosides on the surface of neurons that are associated with the tetanus toxin receptor. Once bound, the tetanus toxin is internalized and then transported from the nerve termini to the cell bodies and the central nervous system. Here, the toxin can reach inhibitory neuron terminals. Let's look at this schematic to understand the path of the tetanus toxin. First, we have the injury at the skin, which leads to the tetanus toxin entering the PNS neurons in blue and traveling to the spinal cord where it encounters interneurons in black. Before we continue, let's look at some of these interneurons. We will focus on GABA and glycine interneurons since the tetanus toxin targets these. In normal conditions, glycine and GABA act as inhibitory neurotransmitters in the CNS. The channels that you see here are where glycine and GABA bind respectively to trigger the release of chloride across the membrane into the postsynaptic cell. The influx of negatively charged chloride inhibits postsynaptic cells. Therefore, GABA and glycine act as inhibitory neurotransmitters and prevent an action potential from firing down the motor neuron and thus induce relaxation of the muscle. But in non-normal conditions, when the tetanus toxin is introduced to the bloodstream, GABA and glycine function are affected. Let's pick up where we left off. The tetanus toxin is using the PNS neurons for transport to the spinal cord. In our schematic, this is when the PNS neurons, blue, meet the interneurons, black. We'll name this synapse 1. Let's zoom in to figure out how the tetanus toxin targets GABA and glycine interneurons. Once in the spinal cord, the tetanus toxin has a choice. Which neurons should it spread to next? Before the toxin can reach its end goal, it encounters excitatory and inhibitory interneurons in the spinal cord. It preferences inhibitory interneurons, GABA and glycine. But how? As the tetanus toxin searches for its next neuronal target in the spinal cord, it targets a specific receptor. Ye and his team found that this receptor is SV2. But we run into another problem. Both excitatory and inhibitory interneurons in the spinal cord has SV2 receptors. So we ask again, how is the tetanus toxin's preference for inhibitory interneurons established? The answer lies in the two orthologs of SV2. It turns out that SV2A concentrates to GABA and glycine inhibitory interneurons, while SV2B concentrates to excitatory interneurons. Importantly, the tetanus toxin has a higher affinity for SV2A and therefore GABA and glycine inhibitory interneurons. This is how the tetanus toxin targets inhibitory neurons over excitatory neurons. Once the tetanus toxin targets the SV2A receptor, it binds and enters. In a normal presynaptic cell, the vesicles release neurotransmitters and intermingle with the membrane before they reform and recycle. During the point of intermingling, the SV2A receptor is exposed to the synaptic junction and the tetanus toxin binds. Then, when the synaptic vesicles are recycled, the tetanus toxin enters the inhibitory interneurons. Once in the inhibitory interneurons, the vesicles travel from the termini to cell bodies where they synapse onto motor neurons. Let's recall our schematic. The tetanus toxin is in the inhibitory interneurons, black, traveling to the motor neurons, red. This is synapse 2. Let's zoom in. Here, the tetanus toxin prevents the presynaptic release of GABA and glycine onto motor neurons by cleaving VAMP, aka synaptobrevin. First, the tetanus toxin unbinds from SV2A and localizes to the vesicle membrane. Then it releases its light chain, which diffuses through the vesicle membrane. Once outside the vesicle, the light chain attaches to synaptobrevin. Upon contact with synaptobrevin, the light chain uses its zinc peptidase to cut synaptobrevin and render it useless. Synaptobrevin is crucial for the release of GABA and glycine from synaptic vesicles. Without it, the vesicles cannot fuse with the cell membrane and release GABA and glycine. With the tetanus toxin rendering synaptobrevin useless, there is no release of inhibitory neurotransmitters GABA and glycine. Luckily for the victim, the tetanus toxin does not enter autonomic motor neurons or visceral motor neurons. Therefore, the tetanus toxin only indirectly affects skeletal muscle, while smooth and cardiac muscle remain unaffected by the toxin. But why? Because the toxin only targets the SV2A receptor, this receptor is only present on GABA and glycine interneurons. GABA and glycine interneurons only directly synapse onto motor neurons, in particular somatic motor neurons, which only synapse onto skeletal muscle. If this specificity did not exist, the tetanus toxin would affect all muscle types indirectly and the victim would perish. So, as the tetanus toxin spreads, it searches for the SV2A receptor and binds, preferentially targeting GABA and glycine inhibitory interneurons in the spinal cord, which is why the tetanus toxin only affects somatic motor neurons and, as an indirect result, the victim's skeletal muscle. Therefore, it is through an indirect effect that causes the tetanus toxin to target somatic motor neurons. This is why we see a rigid paralysis induced along with the spasms characteristic of tetanus, but no effects on the heart or other organs.
As we mentioned, the lack of inhibitory neurotransmitter release, specifically GABA and glycine, lead to hyperactive motor neurons. But why? Tetanus toxin specifically blocks the release of inhibitory neurotransmitters from inhibitory interneurons, but it does not impede the release of excitatory neurotransmitters from excitatory interneurons. In tetanus, only these excitatory signals reach motor neurons, causing them to continuously receive signals to fire off action potentials to skeletal muscles, causing contractions. The motor neurons do not receive glycine inhibition signaling from the impaired inhibitory interneurons, so the firing of motor neurons is never inhibited, and skeletal muscles are continuously stimulated. The lack of inhibitory signaling from the interneurons leads to overstimulation of the skeletal muscles and tetany. According to Cleveland Clinic, tetany is a series of involuntary muscle contractions from prolonged overstimulation of motor neurons. The involuntary control and lack of recovery due to the suppressed inhibition leads to eventual spastic paralysis of the muscle. Unlike flaccid paralysis, where the muscles are not receiving signals to contract, spastic paralysis is marked by stiff and rigid muscles that spasm occasionally involuntarily. From analysis done by Penn State, tetanus is typically known as lockjaw due to the characteristic spasms and locking of the jaw one experiences when inflicted with the tetanus toxin. But it has the ability to affect any skeletal muscle in the body due to how it spreads throughout the nervous system. Skeletal muscles include a whole slew of muscles, from muscles in the face, abdominal muscles, muscles in the limb, and muscles in the neck and back. When inflicted with the tetanus toxin, all of these muscles have the possibility of experiencing overstimulation and paralysis depending on what inhibitory interneurons are affected by the toxin, what motor neurons would normally receive signals from these interneurons, and what skeletal muscles receive signals from those motor neurons. When the affected interneurons are ones that signal to motor neurons that control skeletal muscles found in the back, it leads to a condition known as opisthotonus. Opisthotonus, as defined by StatPro's article on opisthotonus, is characterized by a severe backward arched posture due to the overstimulation of skeletal muscles in the back, neck, and lower extremities. Continuous, uninhibited contractions of these muscles lead to this contorted posture. The painting on screen by Sir Charles Bell illustrates this posture while additionally highlighting other uncontrolled contractions tetanus toxin promotes by inhibiting GABA and glycine release in the inhibitory interneurons in the signaling pathway of the affected muscles. Note the contracted arms, hands balled into fists, the clenched jaw, and contracted toes. These episodes of intense involuntary contractions can cause tears in the muscles, muscle weakness, and fractures due to the sudden contortions and overworking the muscles. While the overstimulation of skeletal muscles that cause opisthotonus have one of the most striking visual effects on the body, overstimulation of other skeletal muscles have further detrimental effects that are not as visible as opisthotonus. The muscles involved in swallowing are skeletal muscles, making the entire process of eating and drinking difficult as an individual suffering from the tetanus toxin will encounter two major difficulties. First, the characteristic clenched jaw makes it difficult to get food into the mouth. Second, swallowing is a voluntary action, but those afflicted with the tetanus toxin may lose control of the neck muscles involved in swallowing, leading to swallowing difficulties and potentially choking. The chest muscles and intercostal muscles are skeletal muscles involved in breathing. While breathing occurs passively, it can be voluntarily controlled, and the contraction and expansion of these skeletal chest muscles allow for the lungs to contract and expand. When the contraction and expansion of these muscles are disrupted, such as when motor neurons that control these muscles have been affected by the tetanus toxin blocking inhibition, the individual may experience difficulties in breathing and may have to rely on a respiration machine until the toxin has run its course and degraded.
With the tetanus neurotoxin binding to the presynaptic membrane of the neuromuscular junction and taken up into the lower motor neurons, the recovery of the synapse is very slow and requires the formation of new nerve terminals in presynaptic structures. With tetanus toxin having a half-life of about 5 to 6 days, meaning that it roughly takes 5 to 6 days for the toxin to become half of the concentration from the initial amount, this toxin can be broken down into the gastrointestinal tract due to the lack of a protective protein complex. This toxin can be broken down into three fragments: A, B, and C. Fragment C is a non-toxic fragment that possesses an immunological feature that helps neutralize the lethality of the tetanus toxin and is thought to be a key candidate for the vaccine against tetanus. The light chain, also named fragment A, has a zinc-dependent proteolytic activity responsible for the synaptobrevian cleavage. The heavy chain is composed of fragments B and C. Fragment B is located at the N terminus of the heavy chain and is responsible for cell toxin penetration, while the TT protein is completed by fragment C, located at the C terminus of its heavy chain, is involved in neural cell binding. Another way that the synapse recovers is through toxin degradation, where multiple variations can occur, but in this situation, the most likely mechanism that would occur is microbial absorption. This is a surface phenomenon where heavy metals are positioned on the cell surface, and through absorption, the inorganic materials are absorbed into the cell. This mechanism consists of microbes collecting inorganic materials by attaching substrates to them and then absorbing them through the gastrointestinal tract. Once the absorption has occurred and all the inorganic materials are gone, it will stop the blocking of release of neurotransmitters at the inhibitory synapse, allowing neurons to once again send signals to other neurons and tissues. A low pH in the stomach prevents microbial colonization, with a pH typically less than 4, altering the chemical form of the toxin, making it more or less readily available to cross the microbial cell membrane. Generally, acidic conditions increase the absorption of acidic toxins, while alkaline conditions increase the absorption of basic toxins, thus impacting the overall toxicity of the substance to the microbe. The combination of tetanus toxin fragmentation and the acidic pH of the intestinal tract, the tetanus toxin is absorbed by the body and no longer exhibits any effects.
This was how the tetanus toxin targets and affects the nervous system. Thanks for watching.